So in that last portion, I was talking about the visceral sensory receptors. So these monitor your cardiovascular, respiratory, digestive, urinary, reproductive systems. So these are receptors of your visceral organs and sending sensory input into the central nervous system. And then the special sensory receptors of our special senses: smell, taste, vision, balance, and hearing. And again, sending sensory input via the afferent division into the central nervous system for processing. So that's the function, the job of the afferent division. Efferent has both somatic and autonomic components, referred to as the somatic nervous system, there you go, and the autonomic nervous system. So somatic nervous system is usually pretty easy to understand. Our voluntary control of our skeletal muscle. So that example of swatting the mosquito on your skin, right? Sensory input, it would in this case be somatic, right? Skin surface. Action potential goes in, afferent division, sensory neuron into spinal cord, brain, processing. A mosquito's biting me, I want to kill it. Motor command output, action potential via a motor neuron, via the efferent division, somatic nervous system, and then that innervates the skeletal muscle, conscious control of the skeletal muscle to try to swat the fly. Okay. The autonomic nervous system, it's different in the sense that it is involuntary. Smooth and cardiac, muscle, and glands are the effector organs in this case. So what do we talk about? You hear the ice cream truck, special sensory receptors, hearing, or you see it come around the corner, vision, right? Or if you're in the mall and you smell Cinnabon, that would be smell. Anyway, sensory input, afferent division, into the brain, processing, food, okay? Motor command goes out, efferent division via the autonomic nervous system, ANS, and that would, innervate smooth muscle. Your stomach starts to gurgle and make noise because of the smooth muscle contraction. Your heart might start beating faster, depending on how much you love that Cinnabon. Your salivary glands will start to produce saliva, right? Autonomic nervous system, involuntary control, things you don't have to think about in order for them to occur, but they're still receiving motor commands from the central nervous system via the autonomic nervous system, okay, which is a component of the efferent division. So it comprises the autonomic nervous system, parasympathetic and sympathetic divisions. They tend to work in opposition of each other. For example, if you smell Cinnabon, right, the sympathetic division will initiate salivation by stimulating the glands. After that has passed, you no longer smell Cinnabon because you've eaten some. Then the parasympathetic division will slow down or stop the production of the salivary gland producing saliva once you've eaten. So they tend to work in opposition of each other. Sometimes these are called the fight or flight division and the rest and digest division, because those are the basic reactions that occur. The sympathetic division tends to prepare your body for emergency type situations, fight or flight. Think about, exiting a classroom and you turn a corner and you see your ex standing there. Your heart rate's going to go up. Your skeletal muscles are going to get more blood and glucose for a potential response to that potentially perceived emergency type situation. The opposite of that, rest and digest, let's say you've had a big meal, blood has been diverted from your skeletal muscles to your digestive tract, smooth muscles. That's one of the reasons why you feel lethargic after you eat, and you're focusing on digesting food and saving energy. So these tend to work in opposition of each other. the sympathetic and parasympathetic divisions. Okay, let's look a little bit at the cells of the nervous system. Two types of cells, if you remember, neurons and the supporting neuroglia or glial cells. So neurons, these are specialized cells. They have excitability in the sense that once they're stimulated to the proper extent, they will generate an action potential. Okay? The stimulus depends on the neuron. If it's a neuron in your nose for smelling, olfaction, the stimulus is going to be chemicals in the air, odorant molecules that would land on receptors on those smelling cells. And if they latch onto some molecule that can elicit an action potential, that signal, action potential signal gets sent into the central nervous system, the brain, via the afferent division. Afferent pathway, your brain would interpret a smell based on memory as a rose or cinnamon or fried chicken, whatever it might happen to be. Okay? So excitability means that the neurons can get stimulated, excited, and generate an action potential. So the neuroglial cells, or the neuroglia, are the supporting cells. And you have a lot of them, about 100 billion in total, about five times as many as your neurons. So that means we have, about 20 billion neurons that actually do the integration, processing of information, receiving input, and sending out motor commands. But you have many more neuroglial cells. And your figure 13.5 tells you what they do. So some of the basic functions, they help isolate neurons, they provide a supporting framework, they're phagocytic, immune system type of function. So the neuroglia in the central nervous system are four types. So notice in the table here, there are 4 neuroglial cells in the central nervous system, meaning within the brain and the spinal cord. And then there are 2 out in the peripheral nervous system in the nerves. So the first type of neuroglia in the central nervous system are the astrocytes, because they kind of look like a starburst, star-shaped, with the extensions. And they're important in maintaining the blood-brain barrier. They have extensions that envelop the capillaries in the blood, in the brain, and they function to provide a barrier regulating the things that leave the blood vessels, like oxygen, glucose, readily, easily, other substances not so well. So they help maintain the intracellular environment around the neurons, which is very important. So the astrocytes help form the what's called the blood-brain barrier. There we go. Oligodendrocytes are the next ones. And what they do is they insulate the axons, the extensions of neurons, with what's referred to as a myelin sheath. So they form a myelin sheath around the axons to help isolate them, insulate them, but also help, as we'll see, with the speed of travel of the action potential down the axon. So they generate the myelin sheath within the central nervous system. Microglial cells, they are phagocytic, so they consume potential pathogens within the central nervous system. And then lastly, we have the ependymal cells. They are unique cells. They have cilia. They line the cavities within the brain, referred to as the ventricles, and within the central canal, which is within the spinal cord. And the cells are stationary. The cilia move, so they move something. They move and circulate the cerebral spinal fluid within the ventricles and the central canal, and allow the cerebral spinal fluid to move, as we'll see in the next chapter or in subsequent chapters. Okay, moving out to the peripheral nervous system, think of like the nerves. We have satellite cells here, and their function is to support cell bodies in what's called ganglia. So what ganglia are groupings, clusters of neuron cell bodies. So that would be a cell body with its nucleus and its extensions, but neurons have also a cell body with the nucleus and long extensions, and sometimes those cell bodies are clustered, grouped together in the peripheral nervous system, and those clusters, those groupings of cell bodies, again in the peripheral nervous system, are called ganglia. So what the satellite cells do is they surround the cell bodies and ganglia and support them, protect them in a way, but again in the peripheral nervous system. And then Schwann cells, also called neurolemnocytes, what they do is they produce the myelin sheath on axons out in the peripheral nervous system. And they also surround other axons incompletely, basically. So they surround what are called unmyelinated axons, which are not completely enveloped with what's called myelin. So the oligodendrocytes and the Schwann cells do a similar function, except in different places. Oligodendrocytes do the myelination in the central nervous system. The Schwann cells do the myelination out in the peripheral nervous system. Okay, so let's look at a typical neuron. The functional cell unit of the central nervous, of the nervous system. Overall structure, cell body, lots of organelles inside. Remember we talked about cells way back like in chapter 1 or chapter 2. What are referred to as nissl bodies are the ribosomes and the rough ER for protein synthesis. They just happen to be called nissl bodies, named after Dr. Nissl. Okay, these darkly staining structures on a slide. There are dendrites, which are extensions coming off of the cell body. And these dendrites are sensory in the sense that the dendrites have receptors for stimuli. Highly branched. So for example, if this is a neuron in your nose that's modified for smelling, the dendrites have receptors for odorant molecules. They land on there, stimulating the cell. Remember, they have excitability, generating action potential, and then the action potential will shoot down this long extension called an axon. So the axon, also called a nerve fiber because they tend to be very long. It's a long process, extension off the cell body coming off of this base called an axon hillock. The plasma membrane of an axon is referred to as the axolemma, the axon membrane. Remember, like muscle fiber, its plasma membrane is called the sarcolemma. Collaterals are side branches off of the axon. Relatively rare. I don't think I'll ever mention them again. And the function of the axon is to conduct an action potential or nerve impulse. That means transmit it, carry it, or propagate it all the way down to the end of the axon. At the end of the axon, it tends to branch, and the branchings are called telodendria. And at the end of the telodendria are the kind of club-like axon terminals, sometimes called terminal boutons, and they contain neurotransmitters stored in vesicles that get released when the action potential reaches them. Remember we talked about acetylcholine being released at the neuromuscular junction? What this figure is showing you, though, is one neuron synapsing with another neuron, which is referred to as the postsynaptic cell or neuron. And you have billions of these neurons synapsed with each other, and one neuron might synapse with 3 or 4 or 5 others, like within your brain and in your spinal cord. So the axon, this long extension that propagates the action potential or nerve impulse, is also called a nerve fiber, again because it's very long. It can be myelinated or unmyelinated. Myelinated meaning it's fully enveloped with a Schwann cell that wraps all the way around. This would be a Schwann cell. So we're talking about an axon out in the peripheral nervous system. And the myelination process we'll talk about in the next lecture.